This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 288, recorded on June 6th, 2014. Hi, everybody. I'm Vincent Racaniello, and you're listening to TWiV, the podcast all about viruses. Joining me today here in the TWiV studio, Dickson D. Despommier.
Hello, Vincent.
How are you, Dickson?
Very well, thank you.
D-cubed.
D-cubed.
I like that.
Yeah, I do too.
You know, Roland R. Rucker, we used to call him R-cubed.
At least I'm not square.
You could have been. Take out the D, right?
That's true. A lot of people have accused me of that, but I said, no, I'm cubed.
Nice weather today.
It's beautiful out. It's a Georgia O'Keeffe sky.
It's got clouds in the sky.
No,
they're not irises or
she
whatever.
Painted, she painted sky pictures also.
Really?
Huge sky pictures with clouds, and they were beautiful, and they looked just like they
It's
do.
22C.
Yep.
Partly cloudy.
Yep.
Very nice day.
Yep.
Gorgeous sun. Also joining us today from southeastern Michigan, Kathy Spindler.
Hi, everybody.
You know, I introduced Michelle Swanson yesterday on TWiV from southeastern Michigan. She didn't like that.
Why?
I don't know. She said it would confuse people, so she wanted me to say Ann Arbor. So, you know, when Michelle says something, I just do it.
Yeah, well, Ann Arbor is in southeast Michigan, so that's fine.
Yeah. So I will call her Ann Arbor and you southeastern Michigan.
And we're right down the hall from one another.
It's okay. Everybody, science is cool because everyone's different.
Yep.
Right. Also joining us today from Western Massachusetts, Alan Dove.
Good to be here.
We call it Western Massachusetts because we don't want to tell everyone where you live. Otherwise, they'd all be at your front door.
Yeah, right. It'd be a bit of a schlep for most of them to get here, probably.
So, Alan, has Mount Snowplow melted yet?
Mount Snowplow is gone.
Oh.
And by the way, Dickson, I just realized that when you lie down, it
Yes.
must be D-cubed decubitus.
You know,
We get a title out of that one day, right, Dickson?
yeah, I'm leaving right now. That's the end of it. I can't do any better than that. That was perfect, Alan. Perfect.
Also joining us from North Central Florida, Rich Condit.
Hi, everybody.
Hello, Richard.
You okay with North Central Florida?
Yeah, I don't care. You know, right? I mean, they do actually call this North Central Florida. That
Yes,
you know,
they
that people understand what that is. It's
Just don't call him Florida man.
Yeah, don't do that.
Why is that? I don't get that one.
Neither
Well, that's
do I,
always
but
the start of a headline about something really
Oh,
bizarre.
yeah. Florida man.
Florida man ran over 3 eagles today while on his way to killing the last panther in
Florida man
Focaccia
electrocuted while urinating on third rail, that sort of thing.
and derails train.
So, Dixon, you're the New Jersey man.
I'm Jersey guy.
Jersey
I'll
guy.
be the Jersey boy. In fact, we both are Jersey boys.
so you need to know that it's 90 degrees Fahrenheit, 32.2 Celsius,
It's getting up
with
there.
a partly cloudy sky.
And humidity?
Jeez.
Yes.
I don't
It's
know.
not a dry heat.
Humidity,
in Florida.
49%.
Look at that.
The dew point
Dew
is
point.
68 Fahrenheit. It's not too bad.
No,
okay?
not too bad.
Not too bad.
It's getting
It's
there, though.
early in the season.
Somebody posted on the TWiV.tv website today, when you guys do the weather, it puts me to sleep.
Wake up!
So I said,
you know what?
So I said, mission accomplished.
That's right. This is not a one-dimensional show. Come on.
I don't know. Most people seem to like it because when they write, they always
I guess
tell
they
us
all
the
put
weather,
They
yeah.
all put
tell us the weather. I think
That's
it's
right.
pretty cool.
That's right.
We have a few follow-up stuffs today. First, two things. I want to announce next week's live streaming event from the ASM headquarters in Washington, D.C. This is called ASM After Hours, and they have events right there in headquarters, which is on N Street. So those of you who live in the area can actually go, and they have speakers and they live stream it. This one is called The Microbiology of Cheese.
Right.
Are they going to take pictures?
Arg.
Say cheese.
Oh, yes.
Arg.
Thank you, Kathy.
Arg.
I'm a bit
Or
slow.
even cut the cheese. No, we won't
Yeah.
go there
Oh,
either.
Dixon. So juvenile.
Of course, they may just have the president there.
Yeah,
The
really.
big cheese. Oh, yes.
The Microbe has published a series of articles about the cheese making, so I'm not surprised that they're doing this.
They have Dr. Rachel Dutton from Harvard University and Matteo Kaler from Jasper Hill Farms to talk about how you make cheese from microbes. And I will put a link. It's microbeworld.org/afterhours. And after hours is all one word. And if you're not there, you could watch it on the live stream, and there's a chat room, so you can put your comments in.
So if you're in the D.C. area, you can go get yourself some culture.
You can.
Bum.
And I think they serve beer.
Oh, cool.
Kathy Spindler, your sigh before reminded me on Twitter earlier today. Our friend Colm wrote another item for the bingo. The Twix bingo board would be Kathy's exasperated sigh at a bad Alan Dove joke.
Perfect.
And so
That
he
is
said,
good.
Perfect.
yes, that's cool. Thank you, Colm. All right, the next is sad news. The Niseen Musache from North Central Florida sent a link to a New York Times obituary of Ciro de Quadros, who was a Brazilian epidemiologist. He was the former head of the Pan American Health Organization, really instrumental in eradicating polio from Latin America and the Caribbean. He died, 74 years old. I met Ciro at a meeting years ago. It was a National Academy meeting. So anyway, I'll tell you that in a moment. But he was a real mover in that area on polio eradication and other infectious diseases. Good stuff. And I met him at a meeting in Washington years ago. I sat next to him. Nice guy. And I had talked about the need probably to switch to IPV to really get rid of polio.
Oh.
And I remember him saying, Vincent, do you think it would work? It's not been tested in these hot areas. I said, yeah, I don't know. That was what I remember of Ciro. I was lucky to be able to talk to him.
Brazil is pretty famous for pioneering the concept of national immunization days,
That's right.
Yes, they are.
where you round everybody up and whack them at once.
Yep.
That's right.
And I wrote Niseen and asked him if Ciro was behind that, and Niseen said he believed so. I can't believe he wasn't at least involved at some level, because that was kind of his passion.
So what happened
That
was
sort of thing.
Saban went down there and collaborated with Ciro and set up these national immunization days and
Okay.
showed that they worked. And that's why WHO figured it would work globally, because of that experience. Yeah.
Right. I also can add to that, because they also are one of the leaders in immunodiagnosis. And the reason for it is because of the addresses of the people. They know almost where everybody lives. So they can actually do gridding of the entire country and find out through the mails where things come from. And so they can actually contact people this way too. And I learned that through this test for Chagas disease, which they actually tested by sending the blood samples on little pieces of filter paper around the country 4 times to come back to the lab and then test it to see whether it would survive being mailed a long time or not. And it did.
Can they find out through the females too?
Alan.
Listen, I wanted to hear Kathy.
You don't look like Alan.
I just wanted to hear Kathy's side. That's bingo. All right. They have Chagas Institute there, right?
They do.
All right. John writes, it's 17C here, 2 miles from Google's main campus in Mountain View, California. Alan's pick for episode 287 was a site called Do Not Link that prevents search engines from following outbound links to bad content. Google already solved this problem in 2005. Any link can be hidden from search engines by using the nofollow attribute. See below. The original intent was to combat spam comments on blogs, but there is no reason a blogger could not use it in the main body of a post. So yeah, you just put your link with /nofollow after it. And
Well, it's rel equals no follow. I was familiar with this,
Oh, it has
but
to be
I
rel
have never
equals, yeah.
been quite sure if it works.
yeah, they say they put that on links that you don't. Rel equals
Yeah.
nofollow. Yeah, that's
Yeah.
right. So you have to put
href equals, and then
Yeah.
your link, and then rel equals no follow. And I think most blogging software, you can even set it so that your
Yeah.
links are done this way, or you can choose whether you want your links done this way. But the reason I like the Do Not Link site is that it goes through an additional layer
Oh, yeah.
And it seems to me, I don't know, just intuitively, it seems to me like it's even less likely to be indexed, but maybe I'm misunderstanding the way the search engines are indexing.
I used Do Not Follow for 2 of the links in last week's show notes.
Right.
That was, one was an article that we didn't like very much.
Yep.
And the other was a no-vax thing that we didn't want to link to. All right, thank you. Anyway, that's for some people who don't want to use Do Not Follow. There you go. Thank you, John. Robin writes, stool and somatic motor paralysis. Somatic motor paralysis can result in stool incontinence by affecting the innervation of the striated muscle of the pelvic diaphragm, which includes the 2nd, 3rd, and 4th sacral nerves. S234 keeps the feces off the floor. That's the way your medical students remember it. Huh? This does not affect autonomic functions that influence the viscera, such as the gut. The thoracolumbar outflow from the thoracic and lumbar segments of the spinal cord is the sympathetic component, and the craniosacral outflow, the 10th cranial nerve, the vagus nerve, plus the sacral segments of the spinal cord, is the parasympathetic component. These two affect the viscera. They can be affected in such conditions as diabetic autonomic neuropathy with resulting dysmotility of the gut blamed for disturbances in the flora, and for diarrhea, and also the absence of pain with heart attacks. That narrative is many decades old, and the gut part could well be outdated. Now, just this morning, excuse me, I got an email from Wink, who sent that original kind of cryptic email. And what he meant, he said, you don't need to read this again on TWiV, but I am. I meant that if others' help is needed to dispose of stool, those others may be at risk for infection. Mm-hmm. So that is totally different, right?
Yes.
Right.
That's right.
There you
Right.
go. And that could... Wait, I didn't finish his email. I shut the darn thing down. Let me see here. What's the last part? This could be an evolutionary advantage for polio. Yeah, I mean, viruses could spread that way. Who knows? All right, Robin continues. Does MERS grow in New World camelids? Llama farms can be found in the U.S. If it does, they can be used as live models.
Uh-huh.
Well, those little animals you suggested?
Gerbils or gerds?
Gerds. Statistics and immunization.
That's not a camelid.
The older part of the...
It's a rodent.
Yes.
I didn't say it was a camelid.
Oh, he did. He said, New World camelids. You said, well, what was that little world? You know, come on.
It was totally different.
It was a disconnect.
I was disconnecting.
It was a segue.
A segmented way.
People would call it other things, but it's unfocused.
Speaking of stool.
The older part of the brain, the reptilian or lizard brain, which includes the limbic system, is non-rational and non-verbal. It is associated with emotions, short time frame, and values, long time frame. The intellect associated with the cortex, particularly in the frontal lobes, is verbal and rational and appears to be in the driver's seat. But what is generally not realized is that it is the chauffeur. It will find rationalizations to defend the positions taken by its lizard brain boss. This is true even in the case of the most sophisticated of scientists, and hence the observation by Max Planck that, quote, a new scientific truth does not triumph by convincing its opponents and making them see the light, but rather because its opponents eventually die and a new generation grows up is familiar with it.
Trust science, don't trust scientists.
Or the more colorful, though perhaps apocryphal attribution, quote, science advances one funeral at a time, unquote. Wow, that's a good episode name, isn't it? Not for this one, but politicians and advertisers understand that the way to change the behavior of the masses is to address their emotions. If any numbers are used, they are cherry-picked, kept simple, and aimed at the emotions. Fancy statistics directed at the intellect will be effective only in those who are trained to digest them. It is like preaching to the choir. Among the rest, the monkey brain will use any statistics provided to the extent that they can support the agenda of the lizard brain. If the objective is to get children immunized, the method is neither a deluge of numbers nor a freshet of rationalizations, but rather a display pictures of sick and suffering children with appropriate narratives. Dr. Dickson's suggestion of videos of children with whooping cough is actually quite a good idea. Wow.
Thank you.
You had a good idea, Dickson.
It was, it's about time, huh?
Okay, Dirk writes, Dear Virus Talkers. Oh, that's good.
Yeah.
Next to the Corona
By
Maps.
the way, I just want to interrupt here. The last
Navajo code talker
correct,
just died. Is
that's
that what you were going to say?
That's exactly what I was going to say.
When, today?
No, it was about 3 or 4 days ago.
Because today, of course, is
D-Day.
Yeah,
D-Day.
well,
I was wondering if we were, as long as we're stopped here, virus whisperers.
yeah, we are.
Virus whispers.
Absolutely. I love it. Let's keep that for a title sometime.
Yeah.
Okay, Dirk writes, Dear Virus Talkers. Next to the Corona Map site, which we linked to last time, there is the website below on different parameters of the epidemic. This is the MERS, of course. From the graph of the case number versus time, there might be some seasonality of the virus. Nothing statistically significant, I guess. Every April, the cases go up. Rainy season in KSA, I believe. So it is worthwhile to talk about the weather. Good.
All right.
I also heard a new Minister of Health was appointed, which could also explain the increase in cases.
Yes.
Nice. Thanks for the great podcast, Dirk. Someone also said that the camels give birth synchronously throughout the year, and I think the beginning of the year is when the camels are born, and so those are the ones that get infected.
Huh.
Right.
Oh.
So that.
So there's a hump of new births each year.
Bingo. All right. Someone else take Stephen.
I'll do it.
Do it, Alan.
Stephen writes, Dear Twivsters, sorry, no bingo points here. First, I agree wholeheartedly with your reaction to the quote-unquote ethical research opinion piece. Even if the level of lab accidents that the authors claim / make up were true, the response shouldn't be to stop the work, but rather to improve biosafety precautions. It's also odd that they talk about what is unsafe today by pointing to a nearly 40-year-old potential incident when biosafety precautions were still being developed. But I have an easy-to-address critique. You were much harsher than called for, responding to claims that the 1977 reoccurrence of H1N1 flu came from a lab accident. Too much was made of the word thought, which is a matter of style, not substance. You criticized the use of a review rather than a research paper, but I think Lipsitch and Galvani chose that paper to make a sly attack on Kawaoka, one of the authors. See, Kawaoka should have known better. That's yet another bad debating tactic, especially since, from skimming the article, it looks like the claim that the virus escaped from the lab occurs only in a caption. The body of the paper is far more circumspect. But it doesn't negate that many scientists seem to think a lab accident is a strong possibility. Thanks to paywalls, I haven't been able to access many papers on the subject, but I can quote from the virology.ws site Origin of Influenza H1N1 Virus post by Vincent, which comments on one such paper. Quote, the suggestion is clear. The virus was frozen in a laboratory freezer since 1950 and was released either by intent or accident in 1977. This possibility has been denied by Chinese and Russian scientists, but remains to this day the only scientifically plausible explanation, end quote. I realize that blog posts must rank below even research articles, but I tend to trust the author of this one. But all joking aside, I'm really curious about what we do and don't know and how much we really can know based on the analysis of mutations. I'll admit I'm somewhat skeptical of molecular clock analysis, probably because I'm more familiar with its misuse in linguistics. Historical linguistics often borrows from evolution, sometimes successfully. I get that the similarities between 1977 H1N1 and virus from the 50s suggest that something odd was going on. But I have trouble, as someone outside the field, figuring how much can be hung on that fact and how much is just speculation. In another example, a recent article in PNAS, Genesis and Pathogenesis of the 1918 pandemic H1N1 influenza A virus, used a molecular clock approach to reconstruct when the Spanish flu's H1N1 emerged and to understand the timing of its predecessors. I read the article, but the suggestion that molecular clock reconstruction explained the high mortality in younger adults seemed very speculative. I had a number of complaints, but mostly I found myself wanting to shout that correlation doesn't equal causation. I'd appreciate if you folks could respond to some of those issues. I imagine a whole episode could be spent discussing the implications of molecular clock analysis and similar studies, but I'd appreciate any insight you can offer if you don't want to do that. I realize this letter is already long, but I wanted to thank you for your great podcasts. I finally caught up on all of the Twix podcasts and have started exploring other science podcasts. I haven't found any that match your depth and expertise when making listeners feel a part of the conversation.
Nice.
Well, so the H1N1, the 1977, I was in Peter Palazzi's lab at the time, and they actually got a sample of that virus and did RNase T1 fingerprinting, which was a crude way at the time to say, is this genome like that of another virus? Because we didn't have sequencing yet. And the fingerprint showed that it was virtually identical to viruses that had circulated in 1950s. So now today, I think, of course, the genome has been sequenced since then. It's quite similar, but not identical. I think there are a handful of differences. So it couldn't have been circulating since then, because there would have been more changes, because we know how much the virus change every year. So that's why it's likely that it came out of a freezer, but of course there's nothing that proves that, so it's pure supposition, right? Although it's the only thing that would explain it, but no one is providing any proof. Nevertheless, that's a long time ago, and security is certainly a lot better than now. But look, let's say they were doing a clinical trial, an unethical clinical trial. I mean, they're purposely releasing this thing, so why would you put the onus on a BSL-4 lab where this thing is totally an unrelated incident? And so that's why I don't think it's a good example to set forth. Now, the paper he's citing about the 1918 virus is actually quite interesting. I wrote about it on the blog, and it does use different kinds of molecular clocks to try and reconstruct the precursor of the H1N1, but they also use some serology that I think bolsters the point. It's not just the evolutionary analysis. And so I would refer you to the blog post, which I'll put in the show notes. Take a look at that, and we might get to it at some point, but there's so much that we can do that we might not get to it. But I think it's not just evolutionary analysis. All right. Anything else from anyone?
Yeah, I was not present for the discussion of that article. However, I have discussed it with others, and one of my strongest reactions was the use of the image of the Nuremberg Code of Ethics. Am I the only one for whom that triggered visions of Nazi Germany?
No.
Okay. So the implication of that is that people doing gain-of-function research are a bunch of Nazis. And I had a lot of trouble with that.
Yes.
That was really beyond the pale.
Well, I can't speak to the motivations of the author, as I suspect that that was not the intent. I suspect the intent was that they just went fishing around for some kind of legal framework that they could sort of half-arsedly cite, and that's what they came up with, even though it doesn't really apply.
Well,
But yes,
it really stuck in my craw.
yes, it does. It does great.
All right. The next thing, and now that you're back, Rich, I just wanted to point out, we had talked a couple weeks ago about getting rid of the last remaining smallpox virus stocks. I put a poll up on the blog, and I just wanted to point out that we have 534 votes yes to destroy, 65 votes no, 376 votes.
My
Not
goodness.
not this year, but in the future, 88 votes.
Huh.
And there are quite a few comments there. There are 19 comments right now, and some of them are quite passionate one way or the other, so interesting to look at.
Yeah, I thought this was an interesting poll and an interesting discussion. It'd be interesting to do the poll, with a different cross-section of the population, because, of course, this is reaching out to TWiV listeners.
Yes, of course. And we have a letter later that relates to that, so we'll save it for then. And that brings us to today's paper. Wow. That was quite a bit of follow-up. Today's paper is in Scientific Reports, which is it's Nature's open access journal, right, Alan?
Yes.
Scientific Reports. It's called Characterization of Cytoplasmic DNA Complementary to Non-Retroviral RNA Viruses in Human Cells. And Kathy has volunteered to read the authors.
Akira
Nice going.
Shimizu, Yoko Nakatani, Takako Nakamura, Atsushi Jino Oe, Osamu Ishikawa, Jeff Beck, Yasuhiro Takeuchi, and Hiroo Hoshino.
All right.
Mostly from Gunma University, also Johns Hopkins, and University College London.
So one correction. Jeff Bookie.
Oh, I didn't know that. Okay.
Yeah.
I'd heard it back in the past. Okay, Bookie.
Jeff Beck is a guitarist.
Yeah.
This is all true.
All right, so
Kathy, do you have some special expertise that allows you to do that?
We have a very international department, and I just wanted to know because I had never heard of Gunma University was the main thing that started it.
Uh-huh.
So,
Okay.
Thank you, Kathy. I didn't command her, Dickson. She actually volunteered
Cool.
to do that.
Cool.
Yeah, but yes,
No, it's done very well, by the way. I
I'm
must
very
say.
bad at some of these pronunciations.
This is true.
All right, this is a paper about DNA copies of RNA viruses found in infected cells. And this is a topic that we have talked about before here on TWiV. It was a paper out of Zinkernagel's lab, Rolf Zinkernagel, a number of years ago, which showed that in cells infected with the arenavirus, lymphocytic choriomeningitis virus, which is a negative-stranded RNA virus, they could detect DNA copies of the genome in the infected cells, and that DNA was present only when the mouse cells that they used also had a mouse retrovirus in them, which implicated reverse transcriptase in making the DNA. We've also discussed since then papers where short pieces of DNA integrated into the cellular genome of a number of different animals have been found complementary to a variety of RNA viruses, not just retroviruses, of course, which encode reverse transcriptase, but filoviruses and bornaviruses, Rio, even influenza virus. There have been a number of papers on those. So this paper caught my eye because, not only because we've talked about it, and this provides a potential mechanism, but when I was a graduate student many, many years ago, a paper was published in PNAS by Robert Simpson, who at the time was at Rutgers, and a Japanese author called Iinuma. And the title of that paper was that they had identified infectious DNA copies of respiratory syncytial virus DNA in cells. They call it actually proviral DNA, which is the term we use for an integrated retrovirus DNA in cells. All right, so this was a big deal, and a review article was written in Nature saying this could explain a lot of persistence of RNA viruses like measles and also autoimmunity, et cetera, et cetera. And by the time I was ready to leave, my advisor, Peter Palese, he said, this turns out to be fraud. It's all wrong. The guy made it up.
Oh, dear.
Okay, so that was never reproduced. I looked in the literature, and no one could ever reproduce that finding. However, they may not have been entirely wrong. I don't know if there's infectious DNA copies, but most likely there is some kind of DNA. So
Even a broken clock is right twice a day.
that's great. Where do you get all these things?
Oh, you've never heard that one before?
No, I haven't heard that one.
Yeah.
Can we use that as the episode title?
I don't know how relevant it is.
So in this paper,
So can I interrupt for a moment? Too?
No,
because
you
as a
can't.
non-virologist in this crowd,
No.
have you said bits and pieces of DNA are
Yeah.
found throughout the genome. Have you ever found an example where you could add up all the bits and pieces and get the virus?
No. Typically in one cell, there's a fragment.
Yeah, but in another cell, there might be another fragment. In another cell, there might be another fragment. If you add it all together, you might get the virus.
No, I don't think anyone
No.
has done that nonsensical experiment.
Nonsensical.
I'm sorry, Dickson.
Okay, sorry.
No one's done that.
Just
They're
around.
just pieces, and it's not clear
Yeah, I got it.
what that means at
But
all.
pieces indicates that it was a whole piece of DNA at one point, maybe chopped up and then integrated.
It could have been.
Well, if that's the case, then you should find all the pieces.
I don't think so, Dickson.
So where do you think the piece came from then?
Well, we're going to get to that in this paper.
Okay,
That's
fine.
it. So this paper proposes
All right.
the origin of those
A
DNA
mechanism.
seeds. So what they do here is they use vesicular stomatitis virus, which I think Kathy used to work on.
Yes, I did.
in John Holland's lab, right?
Yes.
There you go. And this is a negative-stranded RNA virus. Infects cells. It makes 5 subgenomic mRNAs from which the proteins are made. And they infect human cells, and then they extract DNA from these cells, and they use polymerase chain reaction using virus-specific primers, and they detect, starting at about 2 hours after infection, what looks like DNA.
So we might want to point out for people who have never done PCR that PCR will only act on DNA.
Right.
So if they amplify something, that means that there was DNA there.
Correct. Now, if you were looking for RNA, you'd include a reverse transcription step to make DNA, right? And actually, in this experiment, they actually treat the extract with either RNase or DNase before PCR, and they show when you treat with RNase, the signal is still there.
Right.
You treat with DNase, there's no more signal, so looks like you're amplifying DNA. Okay, so the suggestion is there's some kind of DNA copy of some part of the VSV genome in these infected cells. And you start to see it at about 2 hours. All right, so this DNA is primarily found in the cytoplasm of the infected cells. They fractionate nuclear in cytoplasm and do the PCR assay, mostly in the cytoplasm. And that's, of course, where the VSV RNA synthesis occurs. And the control for that experiment is globin, and it's mostly in the nucleus, as you would expect. Mm-hmm. And then they ask, what does this DNA look like? So they use primers from various parts of the genome. This long genome encodes the 5 proteins from one end to the other. They're called N, P, M, G, and L. So they use primers for N only, and they can detect N DNA very readily in the cells, and they make dilutions so they can see how sensitive it is. They use primers for the junction between N and P, and you can also detect that, but there's less, because as you dilute it out, you get less of a signal. You can find the L gene mRNA, but you don't find the L, the sequence at the very end of the genome, which is called the L trailer. So they say these look like discrete pieces, and they also use other primers that would amplify other parts of the genome, and basically they amplify things that look like they're DNA copies of mRNAs. So the idea is that the mRNAs are being somehow copied into DNA.
If that's the case, however, how come they get the NP junction piece? Isn't the NP junction piece, the idea is that P is one RNA and N is another?
Yeah.
So, yeah, maybe the bulk of them look like messenger RNAs, but
there's maybe there's some plus-stranded, right, anti-genomes.
It would amplify anti-genome. That's the discrimination that they're trying to make. So It looks to me like there's potential for some anti-genome being there, but maybe the bulk of it being message.
I think that's fair. Yeah. They also want
they have other experiments later on to back up the notion that it's message.
They also look at the polarity of the DNA product, and this is hard to do by PCR because we've been trying to do this in the lab. And even if you design primers that are specific for a plus or minus strand, after the first round of amplification, they're no longer specific. So what they do is they make primers that are specific for the plus or minus strand, and they do a one-way extension. They don't actually amplify it, but they allow the primer to extend on the template, and then they cut it with a restriction enzyme that is predicted to cut up the DNA that would be made. And then you look to see if there's a product after PCR amplification after that step. Okay? And so basically it looks like the polarity of the DNA that is being made is negative stranded, because with that negative strand specific primer, cutting with the restriction enzyme destroys the signal. All right, so it's negative polarity, the DNA, so it's complementary to the messenger RNA, basically. Then they look in a whole bunch of different human cell lines. They infect them with VSV, and then they ask, do we get DNA? And not all of them make DNA. There's a lovely figure which shows that some of them make a lot, and some of them make less, and some of them make none. At least it's not detectable. And I notice my favorite HeLa cells are among the ones that do make DNA to VSV mRNAs. I presume they checked that these were all infectable, right, Kathy?
I'm not sure that they tested that directly, but one would hope.
Oh, they said the time course of... no, that's a different one. I wonder if they looked at CPE or something.
Yeah. VSV is pretty promiscuous, isn't it?
Yeah.
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 Okay, so then they take one of these cells that doesn't make VSV DNA after infection, and they introduce into it a plasmid encoding what's called a line element, LINE, and in particular line 1. Line stands for long interspersed nuclear element, which is a kind of mobile element that's found in all of our cells in our genome. We have many, many kinds. In fact, 20% of our genome consists of line 1.
Wow.
So it's a retrotransposon. It basically encodes 2 proteins, one of which is a reverse transcriptase that is actually active in human cells. And the line elements are believed to be they're very old, and they're believed to be the precursors of retroviruses. And so the idea here is that this could be reverse transcribing the VSV mRNA. So they introduce line 1 on a plasmid into a cell line, the NP2 cells that doesn't make VSV mRNA, and then they infect VSV DNA, and they infect them with VSV, and then they can see that now when line 1 is present, the cells will make DNA copies of VSV mRNAs. So they have very nice controls for that. I think that's pretty convincing. So line 1 can copy VSV mRNAs, although we don't know if that's actually the element in the other cells that's doing it.
I don't know where is the appropriate time to insert this, but the number of DNA copies per cell that they see is vanishingly small.
Of what, line 1?
Of the VSV.
Yeah.
So the highest is in the RCM1 cells, and I think they said that was something like 4.6 copies, and most of the rest of these are really, really low.
Yeah.
So.
Yeah, that's true. It's really low. I mean, there's a lot of line in DNA. 20% of the genome. So there should be correspondingly a lot of reverse transcriptase. Actually, they do a Western blot here. So if you look at 293 cells, which is what they're using for most of the experiments, there seems to be not very much reverse transcriptase in there. They have an antibody against one of these line elements, right?
No, but it's against the binding protein, and they comment on that in the discussion because they see a lot more of the VSV DNA being made in these 293 cells where there doesn't seem to be much of the line element binding protein, but they make a point of saying, but that's not the reverse transcriptase. So maybe this isn't a good representation of how much is there. Yeah,
the line encodes this binding protein as well as the RT. So you're saying the antibodies against the binding protein, yeah.
Right.
Okay. So that's not a good correlate at all, of course, because it could be totally different.
And one thing I wondered about is, there's all these lines, but does the virus have to do something to, is the virus doing something to activate this line reverse transcriptase, or is it, under what conditions is that reverse transcriptase active?
I think it's always active.
So is it just going around reverse transcribing every message in the cell? Well,
not everyone, but these line elements do reverse transcribe our own cellular mRNAs, and there's a very specific way that that's done, and then those DNA copies of mRNAs can go elsewhere in the genome and integrate. So line is responsible for a lot of mobility of other genes. They're called pseudogenes because they have no introns and there's no promoter. But it seems to happen quite readily. They're currently very active in human cells. They're the only active transposons that we know of. So, I think that they're just always making copies of mRNAs. I don't know what the level would be, but since there's so many copies of the L1, it's happening frequently.
But that might also, that might explain to some extent why you get so few copies of VSV per cell, because you've got all these other, the enzyme is busy. I guess you could say there's a long line to see it.
Hey, that would be good. Oof. So the thing that's a problem here is that, or not a problem, but part of the difficulty is that, so the VSV RNA synthesis occurs in the cytoplasm. The line mRNA has to be exported into the cytoplasm to be translated, and that's where the reverse transcriptase and the binding protein would be made, right?
Right.
Mm-hmm.
And then it has to reverse transcribe the DNA in the cytoplasm. And then, of course, if it wanted to integrate, it would have to go back in the nucleus, which does happen, but that could be very rare, that it would then go back in and integrate. So the integration step may be rare.
Right. And this paper doesn't address integration
Not
at all.
It's just looking at DNA synthesis. But they show that the line can do it, but whether or not that's actually what's happening in the 293 cells where they show it, it's not shown.
They've got all these cells that give different amounts of VSV DNA. Did they actually, I don't think I could find this, did they actually assess the line content or activity in those cells to see if there was a correlation?
No, they didn't. That would be a good experiment. I mean, what I think they ought to do is just knock down the line mRNA with RNAi and see if it knocks out VSV DNA synthesis, right?
Mm-hmm.
This looks like an awful lot of metabolic activity with no purpose.
Well, that's a good question. I don't know if there's no purpose. So
that's the point. There has to be a purpose. We just don't know what it is.
I don't know. I mean, look, line elements. I was talking to Steve Goff about this. He said, line elements are parasites. They basically parasitize our genome, and they can exist there. So they don't necessarily have to have a function, but it could be that making DNA copies
in itself, that's
maybe
what
it's
it does.
Useful. Well, the authors say maybe it has a role in innate sensing. Maybe the cells sense DNA, and if the virus is inhibiting RNA-based sensing, maybe if DNA is maybe gets around that, cell gets around that. I don't know. I mean, there could be some reason for this. I don't know. I think that's the interesting part here.
20%, though. I mean, that's an enormous amount of DNA, basically.
They're parasites, Dickson.
I love it.
Selfish DNA, right?
Yeah, exactly.
Yeah, the Dawkins model.
It's like what Kunin said. As soon as you can cheat, cheating will happen in evolution. Remember that, Rich?
So you get one person on a desert island, you have an honest person. You put two on there, and you have a thief.
Then you have a cheater.
That's right.
There are just two more things I want to mention here. So they treat these cells with reverse transcriptase inhibitors, and in fact, some of them do inhibit the production of VSV DNA, which is suggesting that. And these are both 293 and the line expressing NP2 cells.
I had a problem with this. First of all, in a previous experiment, when they did some treatment or other, they also told us that the VSV replication was normal. You got the same titer and messenger RNA synthesis, and they didn't say that for this experiment. And also, why didn't they just test the effect of these inhibitors on the line activity directly, rather than they do some controls showing that these inhibitors work on murine leukemia virus and HIV-1, and that doesn't seem to be as relevant to me as.
So you want to look at line-mediated reverse transcription directly, right? Yeah, that would be good to do. I agree.
It's like your siRNA knockdown experiment.
I think that you could give it a substrate. The way line normally works is really interesting. So the line elements themselves have a poly A tail. So in the DNA that encodes the line, there's a stretch of A's and a stretch of T. And what happens is when line mRNA is made, which happens because there's promoters there, the genome becomes nicked. If the genome is nicked right there, then the T sequence in the line becomes a primer and ends up priming the synthesis on the 3' end of the line mRNA. And that's how you get more DNA copies of line. So it's called template-based priming. And then that line DNA can go integrate somewhere else. So I don't know if that would be happening with VSV mRNA or not. So assaying the activity might be a little tricky. That's all I'm saying there. Then the last thing is they tried two other RNA viruses, echovirus and respiratory syncytial virus. So I just thought this was funny that this is now coming back to that 40-year-old paper. That's the virus they used in that paper, respiratory syncytial virus. And in fact, they do detect DNA. So maybe those guys were right originally.
My goodness.
Simpson and E. Numa. Yeah. The reason this is interesting to me, because I remember Peter saying, this is what not to do in science. I just distinctly remember him, and he's saying these guys are. And Simpson was, his career, he was a young guy, his career was on the way up, and then apparently that just trashed the whole thing, that one paper. But they might not have been wrong.
How about that?
Isn't that, I mean, that's the Spectre forging of the signal transduction cascade and cellular transformation. That was all forged, but he was right.
Right. When
people did the experiment, it turned out that he was right. And then there was another example of forgery, Spanditos and Siminovich. One of them has forged this paper on putting DNA into cells by transformation into eukaryotic cells. It was a complete forgery, but you know what? That works. So it's weird that some of these things, people say, it must be this way. And
Journal of Theoretical Biology would have been a good place for that paper.
But Dickson, I think the question is very interesting. So I suspect if you looked in most RNA virus-infected cells, you'd find a little bit of complementary DNA. Is that just an accident that there's so many lines there, or does it mean anything? I think it's a really hard experiment to do. And that's really the intriguing part of this paper for me.
Well, we talk a lot about the exchange of information between viruses and cells. And this provides some insight into mechanism, at least for one direction of that transfer.
Yeah. All right.
So did we skip over this bit where they make clones of
I skipped over
circularized stuff? Because that's some of the more compelling evidence that they're amplifying messenger RNA.
Yeah.
And I was thinking about this, and I was wondering why you might preferentially amplify messenger RNA as opposed to random chunks or even the whole of either the genome or the antigenome. And it occurred to me that those molecules are packaged differently because antigenome and genome are going to be packaged in nucleocapsid protein, which is really notoriously tough. It really shelters the RNA from all sorts of stuff, whereas the packaging of messenger RNA is quite different. And I wonder if that doesn't have something to do with the selectivity.
Yeah, so the experiment we skipped over basically shows that these DNAs basically they circularized them and sequenced across the junction and showed that they had the poly A tail and the 5' end. So I think the key here is the primer for reverse transcription. As you know, reverse transcriptase needs a primer. If this happens by the normal line-mediated reverse transcription, these mRNAs would have to go in the nucleus and then be primed at a nick in the line DNA element, and the oligo DT of the line element would prime the poly A 3' end. So that would make sense, but it would be a low-frequency event because you'd need to have a nick at a line element. It could also happen in the cytoplasm. I just don't know. But then in that case, I don't know what the primer would be. Because as you saw, the DNAs appear to start. They have poly A sequence at the 3' end. So there probably is some kind of oligo DT priming. I mean, maybe there's some oligo DT floating around the cytoplasm. I don't know.
So can I just ask a naive question? Of course I can. That's the only kind of questions I ask. Is all of the sequence of line in one person all the same?
Yeah, pretty much.
So line 1, there's no variation in it.
All our lines are pretty much the same.
They're all actually
there's probably variation. Yes, there are variations. Of course, from person to person, there's huge variation.
But
and
in the same person, from cell to cell, there's no variation.
No one's looked, but you probably have 1 million base pairs difference between all of your cells, right?
And how big a molecule is the line gene?
Oh, it's like
how many base pairs?
It's 5 or 6 kb.
Really?
Yeah.
Wow,
I think so.
That's impressive. I mean, that's impressive.
You know what, Dickson? This is a very interesting factoid. 1 in 1,000 of human diseases inherited diseases caused by deletions in genes are caused by mobile elements jumping around in our genome.
I got it. Yeah.
So the line-mediated transposition can go into a gene and make a problem.
Right. So there's this new branch of medicine, I'm sure everybody knows this, is that to look around for the exception to the rule. Look for the one that isn't infected rather than all the others that become infected. And you identified in some of the other TWiVs lots of people who didn't get HIV, which they were exposed again and again and again because they're missing something. They have a mutated gene of some sort. And that's the key to understanding the virulence of the organism that's causing the disease in everybody else. So looking around for these line elements, exceptional, maybe not exceptional, I didn't mean exceptional, I mean exceptions to rules, might give a key to a whole set of diseases that we have no explanation for now, but which you could find an explanation for, provided that you did a really thorough study of the line elements in everybody. Has that ever been done?
Well, we have, 1,000 genomes now? You could compare
it would be easy to... No, you're just looking for one gene. You could do the whole United States if you wanted.
Everybody's genome so far has line elements, has line 1 elements for sure.
Right, so you could take everybody's genomes and just do 310 million,
21% of our genome, Dickson.
That's amazing. Doesn't anybody else think that's true?
Yeah,
that's an enormous amount of DNA.
It's the most
of DNA.
It's the most of any of the mobile elements.
That's incredible.
Let's see how many copies that is here.
There's 516,000 copies of line 1, 315,000 of line 2, 37,000 of line 3.
Stop it.
And to answer your first question, Dickson, they're about 6 KB.
Yeah,
That's
6 KB.
remarkable. That's mind-blowing. That's absolutely mind-blowing.
Yeah, it's really cool. But what I think is cool is these are probably the things from which retroviruses evolved.
Also,
Probably.
well, the probabilities look pretty damn good given the odds of the 20% being captured, some of that being captured by another process.
Yeah, I mean, these have probably been in organisms for millions of years, so they could easily have given rise. No, these things are really interesting. In fact, there is a cool review article in Annual Reviews of Genomics,
Yes.
which is open. I think you can see, maybe not. I mean, I can get it from here. That doesn't mean anything, of course. But anyway, it's called Line 1 Elements in Structural Variation in Disease. It's kind of a summary of the disease part of lines, but there's a good basic background in lines. I'll put that link in the show notes.
How far back in evolution does line go?
Really far.
No, say how far. Come on.
All right, I have to get it out of this article.
Give the audience a break.
Hundreds of millions of years.
I mean,
everything has lines in it.
Everything has lines in it.
Yeah. Everything has lines. You can go back through the hominid lineage all the way back into...
No, but I mean down the phylogenetic tree of life.
Yeah, everybody's got transposable elements.
But that's different.
I don't know about lines in particular.
Right.
But everybody's got all kinds of transposable elements.
That's amazing.
This one concentrates on the human ones. No, they're really cool. In fact, there's a meeting every year on transposable elements, and Goff always goes, and he comes back, and he says, "This is the most amazing meeting."
Yeah, I'm sure.
For example, Dickson, there is a line element. Here, let me. This is just great. There's a line element in here, in salmon. It's a DNA. It's not a line element. I'm sorry. It's a DNA transposon, which is different from line, which is basically an RNA transposon. Okay. It's called Sleeping Beauty because it's been it's been reanimated. And there is one in zebrafish.
Right.
What is it? It doesn't have a good name.
I think that. Well, isn't Sleeping Beauty the same? There's a relative of it in a bunch of different species, and it's not just fish. I think.
Yeah, Sleeping Beauty. Yeah, that could be, and they're using it to do gene therapy because it jumps around so much.
It's called Wicked Witch.
It's very cool. We should get a transposon person on sometime.
Yeah,
this is cool stuff because we don't do it justice.
Yeah, well, that's Barbara McClintock's.
She discovered them.
There you go.
For sure. Jumping genes.
She discovered them. That's amazing.
In corn, right, Dickson?
Yes, in corn, yes. It's maize. We call it maize.
It's amazing.
It's amazing thing.
Yeah, see, Kathy got it. She got that joke.
And there was no sigh afterwards either.
No, because it's not typical. All right, let's do some email. We have a lot of email. Rich, can you take that first one?
Sure. So this is a follow-up to the scarification stuff. Clarissa writes, great TWiV. Rich did a great job, and I agree poxviruses are very cool. Clarissa was a co-author on the opinion piece that we did at the same time. Regarding Dick Moyer's paper, you guys were wondering what would happen if, in humans, if vaccinio virus would be injected into someone instead of using the regular scarification route. I can say that the lesions are very, very ugly. Unfortunately, we had an accident in our lab in the past when a student was using a syringe to disperse virus. The needle made a scratch on her finger, but there was some blood coming out of the wound.
Oh, dear.
The whole story is told in the following paper, and she gives a link. I can't say, and if you look at the pictures in the link, they are ugly.
Gruesome.
Graphic.
I can't say that all the severe effects we observed were due to the route of infection because we can't tell the dose of virus that got in her finger, but it was not an injection per se. The needle was used by the student, and she was removing it from the microtube when it scratched her skin, but it was not superficial, such as scarification.
Right.
It's always good to listen to TWiV. I forwarded the link of this episode to my virology class so they can vote. We had a debate last week on this issue. That was whether or not to destroy smallpox. The class was divided in two, and one group was against and the other in favor of destroying the remaining stocks of variovirus. They elected a leader of the group who was in charge of conducting the debate, directing questions to individuals of the opponent group. It was very nice and productive. At the end, they could reveal their real opinion. the majority was in favor of keeping the stocks and reevaluate this issue in the future.
Wow.
Congratulations for the nice work. All the best, Clarissa.
Wow.
Which, by the way, isn't that what happened this round again? With the smallpox stocks, they punted it?
I don't know.
I don't think they announced it yet.
I have not seen an announcement.
I haven't seen it. I thought it was supposed to be May, but haven't heard.
I thought it was supposed to be May. I haven't heard anything.
I just wanted to mention, I interviewed D.A. Henderson a couple of weeks ago.
Right.
And he was telling me that when they first started the smallpox eradication, they were doing the scarification, and they were told, don't draw blood. It's not good if you draw blood. You don't get a good immune response. Mm-hmm. He said, however, sometimes the immunizers would draw blood, and they ended up finding that you got a better immune response when you drew a little bit of blood. Mm-hmm. So I don't know what that means. Maybe just go a little bit deeper. Who knows? Not very quantitative, obviously. Right. But
yeah, they just coming back to the smallpox destruction, May 28th news item from Nature. They did punt. They took it off the agenda on May 28th. They just asked people to vote on an evaluation of this. So they
are they going to do it again next year?
I think probably. This is just going to keep coming up again.
It's like Groundhog Day.
Okay. All right, the next one.
Yes, sir. One other thing on this scarification thing.
Sure.
I think one of the issues here is the extent to which you disturb the skin.
Yeah.
Okay.
Right.
The intradermal injection sort of bypasses that and goes through. If you scarify but go deeply, that's kind of a combination thing in a way. You're putting the virus in deeply, but you're damaging the skin at the same time. And it's that disturbing the skin that apparently, at least the way I interpret this, gives the enhanced immune response.
Good point. As long as you scarify, you're going to be somewhat protected. Yeah.
Which, in fact, is the Moyer Labs experiment, because they scarified and did the intradermal infection.
Okay.
And I just wanted to say hi to Clarissa, number one, and number two, that the paper that she referred to, on which Nassim is also an author, they basically look at the fact that the lab worker had been immunized, but to smallpox, but this had been some years before, and so they used this finding and this lab accident to point out that lab workers may need to get revaccinated.
And my guess is that even if you are freshly if you're freshly vaccinated, you can probably still get a response to a large enough dose of virus, but it's going to be less of a response.
Right. And they make that point too, that this is a pretty high dose.
There's a lot of variation in the regulations about safety precautions for people who work on pox viruses, if you look nationwide. The CDC and the NIH recommend that those are the guidelines, that people be vaccinated, but individual institutions are at liberty to do with those recommendations as they please. And in some cases, people are not vaccinated. In other cases, they are.
Kathy, can you take the next one?
Right. So this is from Dick Moyer. I listened to the TWiV that discussed my upcoming paper in JVI. First, let me say that I was extremely flattered that you chose our paper to present. Let me also say that Rich's presentation of our work was very well done, and that he covered the content very well. Since I know you guys are always interested in factlets, let me provide a little filler discussion. First, why focus on rabbits, as they are both cuddly and relatively expensive? When we first got interested in the field of poxvirus animal models, the good news was that there was a lot of information on various models, which it turns out was also the bad news. The models were terrible, like intracranial inoculation, probably because the models were developed at a time when there was little sophistication and assays were typically feet up, feet down. As the science advanced, the gold standards became better and included a high degree of sensitivity to the virus and attention to models where infections mimic the natural disease, which is smallpox, to me. That led us to focus on the rabbit, which of course, as you now know, is exquisitely sensitive to the virus. However, there's one additional very, very, very important feature reproducibly found in the rabbit, which is not appreciated and generally ignored. That feature is natural animal-to-animal spread via aerosol from ID-infected animals, intradermally infected animals. Right. No fancy device is needed. I needn't tell you that in the event of an outbreak, that aerosol-generated spread is critical. To further expand on this point for 2 more sentences, if I were working on antivirals and wanted to get something through the FDA, I would test any and all antivirals for the ability to block dissemination and aerosol-mediated spread. Mm-hmm. Doses required to block dissemination, which is really the scary feature of the disease, would undoubtedly be lower and hence easier to get past the FDA. Anyway, the rabbit represents nearly the perfect host for smallpox because it mimics virtually all features of the disease, whereas the mouse doesn't. Finally, Rich alluded to other unpublished work. That is true. This was referring to Dick retiring and probably have a lot of work that's not yet published. So he goes on, for the last 2 years before closing the lab, I had the total and complete luxury of doing exactly what I wanted without worrying about NIH. That allowed me some time to investigate what I think is potentially one of the most unexplored yet interesting aspects of host-pathogen interaction, namely what genes of a given host render the animal susceptible or resistant to disease. Without going into detail, we did a huge number of mouse genetic cross experiments with susceptible and resistant mouse strains using cowpox and vaccinia viruses. These experiments were designed to allow us to explore and potentially identify the host genes involved in animals infected by 1, 2 viruses by the same route, and 2, in the case of cowpox virus, host genes involved with 1 virus via 2 routes of infection. We were able to show some fascinating stuff. That is what I am struggling with now in terms of writing it up, because some of the complex mouse genetics complications are really tough to understand and effectively communicate to others. Anyway, that should be coming out later this year. So, Vince, I apologize for this very verbose email, which, when stripped down to its essentials, is a thank you note for me, thanking you and TWiV for your interest in our stuff and for taking the time to discuss our paper on the air.
So, Rich, when Dennis Ruby tested the smallpox antiviral, did they look at aerosol transmission in this rabbit?
I believe that they've used the rabbit model to look at ST246. I can't quote you chapter and verse, but I believe that's been done. I don't know whether what Dick proposes here is true. That is is verified in that model. That would be to do a dose response of ST246 and see if the dosage required to interrupt transmission is different than the dosage required to keep an animal from dying.
Right. Dickson, can you read an email?
I'll try.
This is from Johnny.
Okay. Johnny writes, "Greetings all. This is not a question about viruses, but more fact-checking something I believe Rich Condit said. I am sorry I cannot direct you to a specific episode, but within the first 3 to 4 months of this year. Referring to his own mortality, I believe he made reference to death as returning to equilibrium with the universe, unquotes. I wanted to check if it was something he said. The thought of one's atoms equilibrating with the universe might actually appeal to some as an expression sympathy. Oh, sympathy. He left out that word. The way I remember hearing it spoken conveyed a calm and peaceful sense of rational acceptance of death. This is where having a better grasp and understanding of the laws of thermodynamics would, no doubt, enrich my appreciation of the inherent beauty of the statement. I have to research and review. Thank you all for creating the most informative, engaging weekly program I know of. Your chemistry, and virology, of course, are great. You're better than any old or new radio or TV series ever. Currently it is 12 degrees C with light winds, 14 kilometers per hour, mist and intermittent drizzle in Boston, 11 C in Cambridge. Difference there. Best to all, John Ballinger."
Johnny.
I did say this.
Mm-hmm.
I do think of death as re-equilibrate. Actually, it's I say equilibrate with the universe, or better yet, re-equilibrate with the universe.
Exactly. Become one with the universe.
Right.
We are becoming one with the universe.
I think it's important to spend some time thinking about this so it doesn't sneak up and catch you by surprise.
Here.
You only get to die once. I want to be there.
That's right.
That's an interesting way of looking at it.
And yet you will.
Yes.
Johnny is a doctor in Cambridge,
Right.
and this looks like it's near Harvard Square.
Harvard.
And
That's right.
I get there frequently.
That's
Johnny, we need to go and
Have a drink.
have a burger and a lime Ricky over at Bartley's, okay, and talk about death.
There you go.
We'll do that.
Yeah, you should.
It's-
You're there a lot.
It's probably the most written about subject in poetry.
That's a pretty big deal.
It is a very big
You know, it's funny.
It is, however, death is perfectly safe.
Indeed.
Yes.
Yes.
Once you're there, but getting there may be a little
right.
difficult.
It can kill you.
Oh,
I was thinking, I don't know why this morning on the way in, why did life evolve to be finite, at least on our planet? You know, every other aspect of life is amazing that it works, right? And everything works together and gene expression is regulated so precisely. Yet it fizzles out.
Vincent, it's about recycling.
Just a minute, Dickson. And it's all because of telomeres, right?
No, but if you
So
didn't-
why couldn't they fix that problem?
if you didn't recycle, where
Who's
would all
they?
the life come from?
Evolution.
No.
Yeah.
No, I think you're right, Dickson. I think the planet would become overcrowded, and
That's right.
and in other systems where this has been tried before, it probably didn't work, right?
You know, this is a great segue to my pick of the week.
well, we're not doing.
I know
picks
we're
yet.
not, but when you get to
Well-
it,
Okay.
you will see what I mean.
Okay, go ahead.
But
Kathy.
but I would argue that maybe there would be a way for things to be infinite. In the way that perhaps the universe and beyond,
Yeah.
can just keep expanding. And so, I think it's limiting to think about the carrying capacity of our planet, for instance,
Yeah.
as being what's limiting us and why we have to die.
So if you could set up a simulation where things were not limiting, perhaps you could see how far it could go.
Well, maybe what you're asking is for an extended life rather than an infinite life.
But
I'm
we
not-
have to reach the point in our infection cycle where we're capable of getting on to the next host before we
Yeah.
we have an infinite lifespan.
Well, I think on one of the TWiPs I mentioned that tapeworms don't seem to have any aging genes. You can keep transferring them from host to host, and as long as you do that, the tapeworms are perfectly happy. They never get old.
Can you passage microbes indefinitely?
I don't know.
Yeast get old.
You do?
They
Yep.
do.
Yep.
Yes.
Yep. Well, any free-living organism does, I think. But this is a parasite that doesn't need an aging gene because the host has one.
Of course, it's not really fair to compare bacteria because every time they divide, you have
Exactly.
two. It's like having a new person, right? So
Right.
it's not quite fair.
So they have some of the original molecules of life. If you just keep dividing a room in half, you'll never end up with zero. So
Alan, can you take the next one, please?
Sure. Matt writes, "Hi, Vincent. Naturally, I thought of you today with the polio CIA announcement. I hope you're well and thriving. Below is my view on why we should destroy the, I think he means the variola stocks. Thanks for listening, as always." And Matt says, "Yes, destroy the stocks immediately. Our current risk management methodology is decisively inferior. Our closely interconnected systems are collapsing at an increasingly rapid rate. Consider two recent examples of Fukushima and the Great Financial Crash. The top mathematicians in the world assured us that neither of those two events would happen more frequently than once every several million years, i.e. they were 12 sigma events." I'm not aware of that work, but okay. But that was false and it was stupid, but most of the brightest risk management folks don't know why. That portends danger for retaining such stocks. One key point is that these models that say we have a 12 sigma chance or thereabouts of a catastrophic accident from virus laboratory are making the same mistake made in both Fukushima and Goldman Sachs. To wit, proper risk management must take into account that these are complex systems of interconnected and layered probabilities. Additionally, any estimate of a very small risk contains a margin of error. That margin of error is often larger than the actual risk being estimated. When you layer those massive uncertainties, their products must be multiplied for an accurate risk assessment to be made. These clowns are off by orders of magnitude. They do not employ what is known as meta-probability analyses. Simply stated, we are too stupid as a species at the present time to continue research with smallpox. The foregoing analysis is informed by pages 91 to 98 of a seminal new text on risk management to be published next year. The text, entitled Silent Risk, is available free of charge below. Virologists need to understand this so we don't have a viral Fukushima. And provides a link to the book.
Okay.
All right. Well, that's one view.
Yeah, that's one. There are many more on the website, so check that out. Next one is from John, who writes, Amgen's oncolytic herpes virus improved overall survival with P equals 0.051, and he provides a link to what I believe is an Amgen press release. Amgen provides update on phase 3 study of telimogene leherparepivec. Forget it. In patients with metastatic melanoma. It's LAHERP, leherparepivec. Man, you need some vowels at the end there.
I don't know. I don't know what these people are thinking when they name these things.
Well, there are big problems with drug names. The FDA regulates what you can and can't call things, and then you've got to have something that's memorable but descriptive and will resonate with physicians but not scare patients. And I've actually heard whole talks about this problem, and it turns out to be really, really tough to come up with a drug name.
Yeah.
That's not also a car name, for example.
Yeah.
Yes. Given this mild side effect profile in comparison to chemotherapy, I hope the FDA will find it within itself to overlook the 0.001. Okay, so anything over 0.05 is not good?
Well, that's the cutoff that
for
people
this test,
use. Yeah.
Usually, yeah.
Actually, there were more serious adverse events for TWiV than the comparator drug. And he quotes, serious adverse events occurred in 26% of this unpronounceable herpes virus patients and 13% of GM-CSF patients. Well, these things are really moving along, and
This, by the way, is a herpes simplex virus type 1.
Yep.
Which is the virus that causes cold sores, and it's been crippled by knocking out a few herpes virus genes, so it shouldn't be able to replicate on its own. And it's also had a cytokine GM-CSF inserted into it, which sort of is an immunity boosting function. So it's a pretty common theme in these. A crippled virus that will also boost the immune response. The idea is to get it in the tumor and boost the immune response and have the immune response attack the tumor as well as the virus.
Mm-hmm.
Right. And it also has an insertion, evidently, of a gene to make it more preferentially replicate in tumor cells than in non-tumor cells.
Right.
So it's injected into the tumor, and so it replicates in the tumor cells, causing lysis, and that releases things like GM-CSF, which are pro-immune. Mm-hmm. So, thank you for that, John. And we are back to Rich for Shane's.
Okay. Shane writes, hi, TWiV guys and gal. I'm on my third listen through all of the TWiV episodes. Whoa.
Wow.
I'm up to 153 and have a couple of questions and a comment. On the subject of using an engineered zinc finger nuclease to knock out CCR5 in HIV-infected T cells, will knocking out the gene for CCR5 in a cell only affect its daughter cells, i.e., haven't the receptors already been made on the surface of the cell? 2, aren't the T cells of an infected person already mostly infected with latent integrated HIV provirus? If so, won't knocking out the entry receptor be pointless? So let's deal with those before we go on. Will knocking out CCR5 only affect the daughter? Well, it's expressed on the surface of the cell that you're knocking out, yeah, but they turn over. Okay, so I would expect that the initial cell would eventually wind up being null, but I'm more interested in the daughters anyway, establishing a lineage of CCR5 negative cells.
Well, the zinc finger requires some repair to go on, right?
Yes.
Okay, so that cell doesn't have to divide in order to get the effect. So, yeah, there's a turnover. If you can knock the gene out, then you should already be getting an effect at some point, depending on what the half-life of the protein is and all that on the surface, right?
Exactly.
Right.
And the half-life of the cells that you put in it. Kathy, you had some comment on that, right?
Right. So they're infusing these modified T cells.
Yeah.
And so those evidently have an estimated mean half-life of 48 weeks in this study. So.
What you would hope ultimately is that somewhere in that population is going to be a cell with some stemness to it. I don't know if there are longer-lived cells in that population.
I don't think there are. But that's a good problem because eventually they're going to get exhausted.
Right.
Yeah.
So that's even a bigger problem, I think, than the T cells of the infected person already infected with latent HIV provirus. So in my mind, if you could do this in a fashion where you infused in a person a CCR5 knocked out cell that, and we've done a paper that addresses this to some extent at least, that has some longevity to it, then since the HIV-infected T cells are going to die eventually, you would hope that the CCR5 knockout cells would take over the population eventually.
Right.
But if they've got a limited lifetime, then you're going to need at least repeat therapy or something like that.
Well, as you said, the long-lived T cells, they're always going to be producing virus, no matter whether you knock out CCR5 or not. And that's the problem, because if you're on triple therapy, most of your circulating blood T lymphocytes are not going to be infected, but the long-lived ones are, and that's the problem.
On another topic, my sister has been diagnosed with multiple sclerosis and is considering undergoing monoclonal antibody therapy, but is very worried about the side effects she has been warned about, particularly progressive multifocal leukoencephalopathy, PML. Notice how that just rolls off my tongue, due to reactivation of JC virus. She has been tested for JC virus and doesn't have it, but if she were to undergo monoclonal antibody therapy, is there a real risk that she could possibly contract JC virus and develop PML, even though she does not currently have the virus? She works in a pharmacy, so she sees a lot of sick people. Let's see. So that's a separate subject. Let's deal with that. So as background, JC is a polyomavirus, that small naked capsid circular double-stranded virus that basically exists in a large fraction of the population and happily resides persistently in the kidneys, but under certain specific conditions of immunosuppression, including this monoclonal antibody therapy or notably an HIV infection. The JC virus can blossom in the brain, and these are modified versions of JC, and cause a progressive neurological disorder that is, I think, almost 100% fatal, PML. And this has been an unfortunate side effect of this particular monoclonal antibody therapy for multiple sclerosis. And Kathy, you looked into this, right?
Right. I consulted the National Multiple Sclerosis Society page and also Wikipedia, and I've heard about this a lot because it has to do with viruses in the brain, and this monoclonal antibody therapy seems to alter adaptive immune function. Well, primarily, first of all, it's an antibody directed against an integrin, and so its function there is to reduce the ability of inflammatory immune cells to attach and pass through cell layers in the intestines and the blood-brain barrier. So that's how it's functioning for the multiple sclerosis. But it also has this effect of altering adaptive immune function, both B and T cells, maybe reducing surveillance of T cells in the brain, changing some of the B cell biology and or leading to factors that can increase transcription of the virus. People are trying to figure out the mechanism by which this monoclonal antibody therapy is causing problems. And so to address the question of, yes, even if you're JC negative and you start doing this monoclonal antibody therapy, they recommend that you continue to get monitored for JC because you could become infected with it at any time.
Sure.
Okay.
So I know a guy at Harvard who works on this, Igor Koralnik, and he's a very public-facing physician. So if you want, you can contact him. You can find him online. And he takes care of people who develop PML after immunosuppression. He says 2.5% of people with AIDS, get JC reactivation. Of course, without immunosuppressive therapy because they are already immunosuppressed. So that might be the number you're looking for. Okay, go ahead.
Do you know what the statistics are in the monoclonal antibody therapy?
I do not, but he would know. So, Shane, if you want to find out, ask him.
So, reading on, I just finished watching season 9, episode 18 of the TV series Criminal Minds, entitled Rabid, and thought you might find the plotline of this episode, quote, interesting, unquote. The show follows a team of profilers who track down serial killers. This particular episode involved a serial killer who had a rabid animal bite his first victim. And then he would keep the victim until they developed symptoms, and then have that person bite his second victim, and so on. He would watch them die over a period of months and film the results. They described the virus as having an incubation period of 24 hours, after which time post-exposure prophylaxis was futile. They even monitored they mentioned the Milwaukee case of a woman who was put in a medically induced coma and survived the virus. Yes, that's wrong, dead wrong. The virus can take, depending on actually where you are bitten, among other things, and what the dose of virus is, the virus can take up to months or even as long as a year to cause clinical symptoms, and you can intervene at any time before clinical symptoms appear, and with effect, with a post-exposure vaccination. So
yet more evidence that the entire genre of TV crime dramas has jumped the shark.
Well, I tell you, I just found a paper where they say incubation period as short as 4 days has been reported.
Oh yeah, it can be short. You get a big some dog badly mauls you on the face and neck.
That's right.
And is very sick himself. You can get a huge dose of virus very close to your brain, and it can happen pretty fast. Exactly. So I wonder if this episode, if these people bit each other in the face.
Oh, but of course they did. They were all,
humans infected with rabies. Oh, yeah. I didn't finish it. Humans infected with the rabies virus were making animal-sounding noises and trying to bite everything, which I don't think is very accurate.
Bite everything.
Not quite.
Luckily, I get my information about viruses from TWiV, not Hollywood.
There you go.
Yes.
It is a beautiful day here in sunny Queensland, Australia, 27 degrees C with white puffy clouds. Keep up the great work on your series of podcasts, and am loving that is Vincent's excellent Coursera course. Kind regards, Shane.
Wow.
All right.
Wow.
Kathy, you are on Igor.
Igor writes, hello, Twivers. I suspect you got a dozen or more responses about the CCR5 mutation already, but just in case you didn't, it has been suggested that the Delta 32 CCR5 deletion spread in Northern European population in response to bubonic plague about 700 years ago. And as far as the phenotype, it's been linked to more severe flaviviral diseases, especially dengue.
Yeah, but I don't think we really know what fixed that deletion. These are ideas, but who knows?
Anything in European populations gets blamed on the plague at some point.
That's right. That's right. Dickson, you are next.
Shane writes,
Shane.
Hi, TWiV crew again. As a follow-up to my previous email about MAB antibody therapy and MS, I have a habit of fighting pseudoscience everywhere I find it, and there is a natural MS support group that is advising my sister that dairy products can trigger MS. Quote, dairy is best removed from your diet if you have MS. 2 proteins found in dairy trigger the immune system to attack myelin. Remember, dairy is for baby cows, not humans, unquote. Arg. She did the right things and asked if there are any evidence, which they provided me enough information to do a literature search, and I found these articles, which they seemed to get their information from. I can only read the abstracts because they were, I guess, blocked in terms of the papers themselves, but he gives 3 references. After reading through the abstracts, it seems that they may have a point. I don't know enough about the field of immunology to make a judgment one way or another, but if milk triggers MS like they are claiming, surely it would be more widespread knowledge by now. Cheers, Shane. Any opinions on that one, anybody?
Well, I sent it to one of my colleagues, David Irani, who's an MD, PhD physician who treats MS patients. He's also a virologist. And he says, "Hi, Kathy. I was unaware of the putative association between milk proteins and MS until your email, but this is not something that merits any reasonable level of concern. From a clinical side, I'm unaware of any good epidemiological data to suggest that humans in general, or MS patients in particular, should avoid milk. As an MS clinician, I do not advise my patients to alter their diets in any way simply based on their diagnosis. From a scientific standpoint, the references you cite also hold little sway with me. The studies are old, and the milk protein in question seem to suppress EAE, which is a model of MS, just as easily as it worsened it. There is little evidence that anti-MOG antibodies contribute to EAE or MS progression. At the risk of sounding dismissive, the concern raised basically sounds bogus to me. Let me know if you have other questions or need other information." David.
Did you change that word, Kathy?
I did.
Yes.
Good. I'm glad to hear what he actually thinks.
The word bogus is the word she changed. Nothing substantive about the email.
Right.
All right, Alan Dove.
Okay, Eric writes, "Hello, esteemed TWiV doctors. First and foremost, I'd like to communicate my deep and sincere thanks for all of your incredible work in science and scientific outreach. Hearing experts discuss new and exciting research in the field of virology every week helps to keep me motivated during long days at the bench, in the hood, or behind a desk. I especially enjoyed recently hearing about Dr. Racaniello's formative years, and I hope that Drs. Condit, Despommier, and Spindler will follow suit."
Yeah, let's do an episode.
What happened to Dr. Dove? What happened to Dr. Dove? I want Dr. Dove in there too, damn it. Sure, we could do a
Actually,
solipsistic
that could be
episode.
part of TWiV 300 if you wanted to do that.
I was thinking about that, but then you don't get an episode to each one of you.
Well, and
A whole episode for each one.
A Condit episode, a Spindler.
Holy cow.
I don't need a whole episode. I mean, when you did a TWiV here, you got my background.
Please.
Yeah, I
Kathy,
could do mine in 5 minutes.
I think you need 10 episodes, Kathy.
That complex, huh?
The letter continues from Eric. "I'm writing today seeking some practical advice about something which I'm very much still figuring out. What makes a good scientific presentation? After over a year of hard work, one of my projects is finally ready to share, and I'm happy to say that our abstract was accepted for a talk at this year's ASV.
Wow.
This will be my first oral presentation at a peer-reviewed conference, and although I'm excited and honored, I'm also anxious and filled with questions. So my question for you is simply, what are some aspects of successful scientific presentations? Can you offer any tips specific to ASV? Thank you again for all your hard work. I can't wait to see the live podcast."
That should be another podcast.
Yeah, I have about a dozen or so recommendations. First and foremost, practice.
Right.
Yes.
Make sure that your talk really
much
is
time is
within the prescribed time limit.
What are they, 12 minutes?
12 minutes.
12 minutes, 10 to 12 minutes and 3 minutes for discussion.
That's amazingly short.
And transition to the next speaker. So, yeah.
Yeah.
Do not try and cram all of your data into the presentation.
That's right.
So pick the most important scientific bits that you want to, the most important pieces of data that you want to communicate, and put them on a very simple slide, and then do what I call the figure drill with each one of these, and that is walk through whatever data it is and present very clearly the rationale for the experiment, how the experiment was done, what all the axes on the figure were, what the data actually are, all of that stuff, because that winds up pacing yourself so that people can understand and follow along with what you're doing, and then, and only then, draw a conclusion from that experiment. For any given experiment, that takes a long time. And I think it's a huge mistake that 90% of people make, to get up and put up a slide with a dozen panels on the slide and just draw a conclusion. It's like saying, here's proof that I did the experiment and this is my conclusion. That does absolutely nothing for me. I want to be included in the discovery. I want to know what the experiment was. Don't use a lot of fancy stuff on your slides. White background, black lettering.
Yes.
Times New Roman.
high contrast stuff. Don't use color unless you have to. How am I doing
18-point
so far?
font.
Okay.
18-point font.
Yeah.
And check out.
Go ahead.
I was going to say, check out my pick on TWiV 268. Jean-Luc Dumont has everything slides, how to communicate your science,
Bingo.
how to do the whole thing.
Good.
Many of the things that Rich just said, but if you put something on the slide, talk about it, and if you don't talk about it, leave it off the slide.
Right.
Yeah, and a 10-minute talk is very short. If you're, I don't know, if you're trying to cover more than 2 or 3 experiments, like whole experiments, in that time, then you're doing something wrong. You're really only going to be able to cover a small set of data in that time period and do it with any quality.
Biggest mistake people make is to try and present too much data. And
Yes,
correct.
It just messes things up completely. There's this great compulsion that you want to show everybody everything you did. But if you try and do that, people just zone out.
Well, there's also this confusion that's arisen in the digital age because a video takes so much more internet bandwidth than text. There's somehow this has created the perception that talking is inherently a way to transmit more information, when in fact it's exactly the opposite.
Mm-hmm.
Yeah.
A talk, a presentation, is a very, very low bandwidth thing. The amount of information you can put through there is much, much less than what you can fit in a text document that people can sit down and read through 9 or 10 times.
That's right.
So simple.
I spent 12 weeks, once a week for 12 weeks, sitting at a seminar at Rockefeller University when I was a postdoc there on how to give a 10-minute talk.
Wow.
And at the end, in essence, it was tell them what you're going to say it, and then tell them what you said.
Mm-hmm.
And do 3 points, just 3 main points, and use as many visuals rather than words as you can, and everybody will catch it. And,
There's another presentation website, which maybe I can find the link for, or it's just a video, where it's bullet points kill.
Absolutely.
Instead of having a list of bullet points, put up a picture.
That's right.
And Jean-Luc Dumont, I think, is also the one who says that your visual presentation should make the information completely understandable to a deaf person, and what you say should make it completely understandable to a blind person.
Yes.
So
Outstanding. That's good.
That's how simple it has to be.
Beautiful.
When I live tweet talks, if it's a good talk, I don't have to look at the slides at all.
Right.
Mm-hmm.
I can just listen and look at my keyboard.
Right.
And so, Eric, don't worry, because
no sweat.
Vincent and Kathy and I, if you tell us when your talk is, we'll come and we'll sit in the front row.
Yep.
Okay? And we'll watch very carefully, and we'll make sure you do this right. And we'll give you a full critique afterwards.
So you can relax.
There's no pressure at all.
Nice.
No pressure at all.
Nice, yeah. We'll go. We are going to go. All right. The next one is from Anne. No, sorry. The next one is from James. Hi, all. I just listened to TWiV 279 and thought that you did a good job addressing the question/statement about individuals immunized with measles, mumps, rubella vaccine not being protected from infection with mumps during recent outbreaks. I just thought I would pass around an interesting paper that attempts to model the effectiveness of the vaccine by dose using mumps outbreaks in Canada. The conclusions show the importance of having very high vaccine coverage, especially if the vaccine has lower efficacy in an individual. The rates of vaccine coverage required to prevent community transmission of mumps are very high, using their model with 88 to 98% of people needing 2 doses of vaccine. It is also worth pointing out that while many of the people infected with mumps were vaccinated, most of the cases of measles are occurring in unvaccinated individuals. In both cases, the outbreaks would likely be much more widespread in an unvaccinated or less vaccinated population. All right.
So one of the critical things here is that these recurrences or these new epidemics or episodes of measles and mumps have different epidemiologic etiologies. Okay, the measles infections are showing up primarily in unvaccinated individuals. The mumps infections are showing up in people who've been vaccinated a couple of times and have to do more with the efficacy of the vaccine, more so than in the case of measles, where it's people not getting vaccinated. They're a problem in either case, but they're interesting and they're slightly different.
Yeah, I'm not laughing at that, Rich, at all. I'm laughing at Alan's note in the show notes. I guess Alan wants us to move on. I
just wanted to let you know that we're at an hour and a half mark.
Can we do one more round of emails?
It's fine with me.
Okay, because I think we have lots.
We do have tons of email.
I'd like to get through one. All right, let's do one more round. Rich.
We should do an all-email show someday.
We'll do that.
All email.
Bingo.
All right, we'll do it next week. Okay.
Ann writes, hello, TWiV team. Many thanks again for your engaging and illuminating podcasts. I want you to know that I was inspired by your podcast to sign up for a course in immunology on edX.org, and what I am learning is helpful, helping me keep up with your discussions, as so many immunological aspects of viruses are discussed. The course is a lot of work, but it's been a good course so far, and I hope it will help prepare me for the next iteration of Professor Racaniello's virology course on Coursera. I've attached the link to an article by Ezra Klein, formerly of the Washington Post, and now writing at his own site, Vox. I would be interested to hear your opinions, the opinions of your team, of the assessment by law professor David Kahan, who investigated why people stick to a belief even in the face of evidence that disproves or weakens the argument of the belief. He says that the science community doesn't have a communications team at all, referencing the HPV vaccine rollout debacle. And he gives a link to an article, Brain Dead, How Politics Makes Us Stupid. Many thanks.
Ann is right.
Yeah.
Science does not have a communications team at all. Yep. We do, individual universities do press releases, and this is a big problem. I think if we had a unified, but maybe they wouldn't believe that either. Maybe they think it was fabricated. I don't know.
Yeah.
Right. What do you think, Alan?
Well, I mean, I don't have a good alternative to the current system, because if you try and get everybody to have a unified communication about something, that's kind of anti-scientific in itself.
Yeah. So I think we need individual efforts like what we do here.
Yeah. And the blog post that is quoted here is what we've talked about before and what Robin talked about in the letter at the top of the show, which is that basically people have a greater tendency to follow their reptilian brain, their emotions, than they do their rational brain.
I'm just going to say it's a really good blog post. I think it's well worth reading. I enjoyed it.
Yeah.
Makes good points.
Who's next?
I guess I am.
No, Rich.
Next.
I'm starting with James.
Yeah.
Hello, TWiVites. You mentioned in response to a letter in TWiV 279 how requests for data can be used to harass scientists. This happened on a fairly large scale with regards to Australia's legal wranglings over plain packaging of tobacco products. As part of this, Philip Morris International demanded, via a Freedom of Information request, all data, published and unpublished, including in progress, regarding advertising and merchandising of tobacco products from the Tobacco Research Institute at the University of Stirling in Scotland. This was presumably to hopefully find a smoking gun, sorry for the pun, to support their, at the time, wrangling in Australia. I read, although I can't find the reference at the moment, that the data requested under the FOI request was to include not just raw data, but also all notes made between colleagues, telephone conversations, and interview data. 3 key issues was that the interviewee data was taken in some case from the young, who agreed on the basis that it would only be used for legitimate research, so even anonymized would undermine the promises of the researchers. The second issue was the fact that all the data was being required, there were huge massive work involved in collecting everything. Finally, the major issue was that all this data was being requested before any publications on the specific matter, and they wanted everything from the department. In the end, they dropped the request, albeit very quietly, assumed due to the uproar from the public and the community, so it never got to full legal challenge in the UK. But it creates a bad precedent that it could be attempted. Does anyone know of attempts to actually do so much on such a grand scale? And then he gives a couple of links about this Tobacco Giants' War on Science from the UK in the Independent.
There have been multiple instances of this in different countries. There was, I don't remember the names of the principals involved, but there was one with researchers who were working on the effect of particulate matter on rates of lung disease. And they got harassed by, I think it was the coal industry, that went to similar lengths trying to force them to basically make copies of all of their lab notebooks and this and that, which the purpose of this was just to harass. And it's a legal mechanism for harassment that is available in many countries. There was another one where the paper industry went after researchers who were working on the effects of effluent from the paper industry on lake ecosystems, I think it was. And again, I'm sorry, I don't remember the details of all these, but if you start Googling this issue, you will find other examples. This is not unique.
Well, creationists have had a field day with all this.
They've done some efforts at it. It's mostly well-funded industries that have
the Coen brothers
that have really turned it into a major tactic.
Yeah.
And of course, the NRA actually shut down research at the
right.
CDC for 20 years.
Sure. The automobile industry with regards to safety belts and crash-proof cars and stuff. Yeah.
So, Kathy, James has a PS here. It
Right.
almost looks like another
He
letter.
does. He
Right.
does.
12 degrees C, bright and sunny, with a Beaufort of around 3 outside my work window. While we're on the subject, I think I recall Dickson Despommier fairly recently saying how only America and Britain still use Fahrenheit. I'm afraid that's no longer the case. I'm 28 and I cannot recall a weather forecast even mentioning Fahrenheit since I was in single digits, and even then it was alongside Celsius. In school, Celsius is the only measure used. In fact, the only place imperial measurements are used is legal road speeds and road distances on maps and road signs. Wind speeds in non-scientific environments may be in miles per hour, but scientifically and in the Met Office, knots or MS1 or now standard. Electrical screws are often still based on the British standard screw thread, and occasionally milk will be an even pint instead of an even liter. However, it also has to have the metric equivalent and be sold as such, unless buying direct from the farm door, of course, which would be unpoliceable. So,
Do we know?
And I have
Ellen?
one more, which is that you order a pint in a pub.
right.
Yes.
Okay, you'd never order
right,
a half
right.
liter
No, you would not.
or 500 mils.
So where's
or-
Buford? That's Texas, isn't it?
yeah, but there's
Yes.
a Buford scale for
There's a Beaufort scale.
Wind
Yeah.
speed. Beaufort for wind
I
speed.
think
wind speed.
I think James is from the UK.
I believe that.
Yeah, I think so. And
Yeah, that's
where
right.
he said up raw,
Yeah.
up above, we would think of it as uproar. But yeah, and in North and South Carolina, one is Beaufort and one is Buford, and I can never remember which. So can be pronounced both ways.
Uh-huh.
All right, Dickson.
Ain writes, Dear Vincent, just to touch base on this, I announced the Minecraft virus competition during my talk at the Archer Academy and gave them a link to the new TWiV website so they could get some ideas on virus structures. The teachers at the school will announce the competition formally to the rest of the school, 150 children in total, after the Easter break. They will be given until the last week in May to submit their entries. 3 book tokens are to be rewarded. In the first instance, they will send the entries to me. I will collate them and send them on to you.
By the way, let me interrupt.
Please do.
So they're doing a Minecraft virus competition.
Right.
They're building viruses in the game Minecraft, and we're going to be the judges.
Ha!
All right.
Cool.
Right. The talk to the children went very well indeed. I think there may have been some, no pun intended, budding virologists in the audience. They didn't say that, but I did. Even the teachers said that they were inspired and were surprised that viruses were not alive. I like the way Khan describes viruses on Khan Academy. Left to its own devices, it will just sit there like a book. Thanks for your suggestions. I use them all. By the way, just in case you missed this, did you know that you were cited in Brain on Fire by Susanna Callahan? Now, Callahan.
I think it's supposed to be Callahan.
Callahan,
I
yeah.
thought so too.
Callahan.
Callahan. Okay. I liked your latest podcast on endogenous retroviruses. Mind-blowing. There is so much to know. By the way, pig endogenous retroviruses have also been described in the literature.
You have to say the acronym, Dickson.
PERVs.
Because then she put a little smiley there.
Oh, no, I don't have the smiley.
Haven't we talked about pervs?
Not
Perps.
me.
Perps. I've
Oh,
seen
okay.
perps a lot, but not pervs.
I don't know. I think we actually have.
All right. Best for now.
Right. And since she made the comment about budding virologists in the Holland lab, we all considered ourselves budding virologists because VSV buds out of the cell.
Nice. All right, Alan, you are next.
David writes, Dear Vincent and friends, I'm listening to TWiV 279, and I heard Alan start a thought with, one of the things running around in my head was, and he proceeded to raise the question of how you evolve a system where HERV-H makes itself essential for embryonic development so recently. A discussion followed. But what I really thought Alan was going to ask was whether LNC activity could be part of the life cycle function or of all retroelements, including retroviruses. If so, there could be a whole new dimension to retrovirus interactions with host cells that we don't know about. Thoughts?
Wait a minute. He's careening towards retirement, which I'm sure.
And then, yes, the careening toward retirement, which I'm sure will be made more enjoyable by TWiV.
And, it's interesting. I was thinking of this today because the links, so in TWiV 279, we talked about how an endogenous retrovirus is important for maintaining pluripotency of stem cells. And the way that works is that the RNA of the endogenous retrovirus actually binds at the retroviral promoter and assembles an enhancer complex that is used to turn on the genes that are needed to keep those cells pluripotent. And, the way the lines work is that the RNA that's going to be reverse transcribed goes right to the line element itself and binds there and is primed by a nick, as I was talking about earlier. So it's kind of a similar thing. So maybe there's something to this. So maybe these RNAs are participating in other retroelement functions. So, yeah, I think this will be coming out in the next 25 to 37 years. Yeah.
Given proper federal support.
Yeah, it's a good idea, David. Stay with us. All right, there's one more paper from Robert. Hello, TWiV hosts. I would like to suggest the following paper for discussion on TWiV. Woroby et al., a synchronized global sweep of the internal genes of modern avian influenza virus. I read a short article on this paper in an email I received as a graduate of the University of Arizona. Also read the article in Nature. I think it would provide a good learning experience for the TWiV audience with respect to the phylogenomic methods used in these types of studies. Maybe Harmit Malik could also participate. I would need to have Harmit here to discuss this paper because I could not do it on my own.
It's a tough one. I had a look at it myself. I think it's also quite interesting.
However, I do know Michael Woroby. I have corresponded with him, and perhaps he would like to join us to explain it. So we'll try to do that, Robert. Weather in Orange County today is sunny, 68F, 20C, humidity 58%, dew point 52F, pressure 29.95 inches. We are supposed to get some rain this week with 2 to 3 feet of snow in our local mountains. If this happens, it will be welcome for sure. Thanks for the great show. So Robert is our friend who is a retired biochemist who surfs and skis in the same day.
Wow.
But it's still pretty dry out there. I was there on Wednesday in San Francisco, and there's signs everywhere that say, conserve water. We have a terrible drought.
3-year drought so far.
Here.
3 years.
All right. Dickson.
Vincent, is this all the rest of the email that's on the site here, or is this a bunch you haven't posted?
There's more.
Okay.
There's more.
We should
definitely do an all-email
Yeah,
episode.
we'll do one Friday.
Oh, yeah.
So right here, there's another 50, and then there's another 50.
Good Lord.
I
mean, when it stops snowing, you'll know you caught up. Or maybe it'll be next winter by the time
Dickson,
I
do you have a pick?
I do. It's a great pick, too, and I trust we haven't talked about this yet. There's a book that just came out called The Oldest Living Things in the World. Mm-hmm. You should see this book. I bought it. It's fabulous.
I think you picked that once.
Are you sure I did?
I'm going to check. You go ahead and say it.
No. I told you before, and you said, no, you haven't picked that, so I decided to do it. But maybe I'm just suffering from early onset Alzheimer's.
No, we haven't done it.
Great. Then I picked
so
it.
Are you in this, Dickson?
That's very funny. That's very funny, Alan.
You have a book about the 12 oldest animal. You picked a website which had the 12 oldest animal species on it.
I did.
That was called... I forgot. Anyway.
I hope it wasn't called The Oldest Living Things in the World, because that's the book that was reviewed in Science Magazine just recently.
Actually, that was Rich.
So this is an incredible book because it not only shows you the picture of the organism that they're talking about, they give you world distributions and the ages of things. And there are some organisms out there that she claims, at least, are 100 or 200,000 years old.
What are they? What's
They're microbial mats.
Oh, yeah, the mats are old. Yeah.
I mean, really old. But some other things that you wouldn't think about, like creosote bush, is something like 14,000 years old. That's incredible. So what do they use creosote for? They use it on railroad ties to prevent things from growing on it. So maybe if nothing grows on a creosote bush, it can live a very, 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 very long time. So everybody, listen up. Get that creosote out and use that ointment and spread it all over your body and get rid of that microbiome. And that way you... No, that's not true at all. I'm just making it up. Sorry about that.
This is very cool.
I love the book.
You can see a lot of it on the Amazon site.
I love the book. And Kathy, you're worried about women in science? This is a women-authored book.
Cool.
She is an artist, Dickson.
Yeah, but she's also a scientist in heart because look at the way she presented it. It's a beautiful
Dickson.
book.
She's
Beautiful
an artist,
and
contemporary artist based in Brooklyn.
But beautifully
Her
photographed.
photographs and writing have been featured in the New York Times.
That's right. And scientifically accurate, too, because she collaborated with other scientists.
Thank you, Dickson.
You're welcome.
Alan, what do you have?
I have an experiment called Experiment. This is a site, experiment.com, which domain name must have cost him a small fortune, but this is a crowdfunding site for science.
Nice.
And it's that's pretty much the description. So they've got some featured projects. They've got a little plug from Bill Gates, bunch of stuff about how they work, and it's you've heard of Kickstarter and Indiegogo. This is the same concept, but focused specifically on scientific research. And so you post up your project and see if you can drum up funding.
Very nice.
It's a little tricky with the money because you don't want to pay tax on it, so it has to probably go directly to your institution.
Probably. Yeah, I'm not sure how people are going to handle that. It depends on what country you're in, probably, too.
Yep. Yeah, it's the way things need to go, but, for us...
well, I don't know that it's the way things need to go, but it's how they're going to go, I suppose.
We need to get more money in, but I was just going to say, we can't get enough money in our field by this mechanism to be able to do this.
The stuff that's going to end up succeeding in this is going to be what can grab a lot of attention. Yeah.
And I have a feeling they're going to be garage-type experiments as well. So
that too, yeah.
But it's a good idea.
Phage.
Sure.
Go work on phage.
That's right. Kathy, what do you have for us?
Well, in keeping with the arc of women in science, I heard this story last night on The World that Lego has a competition for new sets. And the most recent competition included one from the fall and then a winter series. There were 7, I think, in total. And the one that won is going to be called the Lego Research Institute. And it was proposed by a woman scientist. And the mini Lego that I got in my Christmas stocking is actually part of this set.
Yeah,
so it's going to be an astronomer with a telescope, a paleontologist with a dinosaur, and the chemist with her lab. And so I think it's great. Just any additional thing that can make people more aware that there are women scientists, that is great.
This is
very cool.
That's the one with the white coat. That's the one you got, right?
Yeah.
It's a little bit different from the one they actually came up with, but yeah.
Cool.
I like the one in the white coat because I like she carries around these beakers.
Yeah.
They're really
That's cool.
And it looks like the paleontologist has a microscope. That's cool.
Yeah.
That's very neat. I saw this today on Twitter. I was thinking about it because I saw your little gal there,
The
Kathy.
the
That's
Muppets
cool.
have Dr. Bunsen Honeydew and his assistant, Beaker.
Yeah.
So maybe you should put those together somehow.
My grandsons, 2 of my grandsons are total Lego addicts, and they brought their Legos with them on a recent visit. And I must say, I can understand the addiction.
Mm-hmm.
Had I
They're
known,
pretty cool.
I would have entered it and put in a vertical farm, but I'll do that next time.
All right.
You couldn't build a Lego, Dickson.
Yes, I could.
Yeah, okay.
Oh, yes, I could.
Rich, what do you have for us?
This I have a preview to. I have lava lamps. So this is I discovered these in Boston. We were at the Quincy Market, and there was a stand selling these, and I posted a picture of it the TWiV Facebook site. These are do-it-yourself lamps that they look for all the world like viruses. Mm-hmm. There are these pieces that you fit together to make a sphere that you can make a bunch of different shapes, but one of the predominant shapes is icosahedral symmetry. And you can get different colors of the pieces to fit together and make them look attractive.
Because they're made from repeating segments.
Yes, as a matter of fact, there is a single repeating unit, as there should be. And actually, the sphere that is the basic icosahedron, when I was looking at the details from that, it said it uses 30 pieces to build it. And I said, wait a minute, that can't be right. It's got to use 60. But if you look at them, they're basically dimers. Mm-hmm. Okay? So it's a 60, in essence, a 60 capsimer particle. And it does indeed have icosahedral symmetry. They're really pretty cool looking, actually.
You will be pleased to know that I did click on the Buy Now link just to see, but all they do is refer you to a toll-free number or their website or an email. And so I called and emailed them and have not yet heard back.
So, Rich, you saw these at the Quincy Market in Boston.
That's correct.
How much were they?
I forget, actually. About $20 or something like that.
Okay.
So Kathy's going to get one.
Yeah, lots cheaper than a Lamborghini.
You're gonna put it at home or in your office, Kathy?
I don't know. I might get more than one if they're that cheap.
Wow. I told Rich to buy a bunch, but he didn't.
I didn't do that.
The George Kovacs lighting company used to make a lamp that you could buy that was a series of flat plastic panels that sort of slid into each other, and they formed beautiful symmetrical patterns also. So. It's an interesting theme.
You know, there's a great icosahedral light from Restoration Hardware.
Is that right?
They have a couple of them.
Oh, yeah.
So I just got yesterday all the Restoration Hardware?
Sure.
For some reason, I got all 20 of their catalogs. They have different ones for like bathrooms and stuff and fabrics.
Sure.
And they sent them all to me in this huge stack.
Wow.
But anyway, they have a bunch of icosahedral lamps. One of them was a pick of mine a long time ago. They're very expensive, but they're beautiful.
Remember that.
My pick today is extremely inexpensive. It is a Facebook website of the American Society for Virology. It's brand new, and we're trying to get it going here. We have 600 likes, so if you like ASV and you want to stay in touch, check it out. Facebook.com/American Society for Virology, all one word. And we have a listener pick from Basel. If you would allow, I'd like to change my pick that I sent in 2 days ago and recommend a different one. Okay. I'd like to pick a treatise on the smallpox and measles by Abu Bakr al-Razi, written in the 9th century and translated in 1848.
Wow.
What amazes me the most in this book is the detailed and thorough literature review Razi provides in his introduction, despite the fact that he didn't have Wikipedia or Google. Also, the eloquent description of the clinical and gross pathology he provides of these conditions is just fascinating. This book is what really got me fascinated in virology and vaccines back when I was in the 5th grade. To this date, I read the book in Arabic back then. It's readily available for free online through many libraries, including Google Books. It's really cool. You get the whole book right here online.
Cool.
That's very cool.
Yep, treatise on smallpox. So there you go, Rich. Pass it around to your
I like that a lot.
pox virus invitational friends. All right, thank you, Basil. And that's TWiV 288, which you will find at iTunes and also at twiv.tv. And do send us your questions and comments to twiv@twiv.tv. Dickson Despommier is at verticalfarm.com. Thank you, Dickson.
Welcome.
And as usual, you don't say anything.
What is there to say after all of this?
You could say, I had a great time. I learned a lot.
Okay.
It was really a pleasure. You used to be more vocal.
I had a great time and I learned a lot.
Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
This was fun. And I forgot to say when we were finishing up the paper that we did that you also blogged about it. And a couple of people put comments where they included some other instances in the literature of that kind of thing. So people might want to check that out.
Yes, I'll put a link to that. Yeah, there are other examples of that now. So it's a growing phenomenon. of unexplained significance.
Yeah.
Rich Condit is at the University of Florida in Gainesville. Thank you, Rich.
Sure thing. Always a good time.
And Alan Dove is at turbidplaque.com. You can also find him on Twitter. Thank you, Alan.
Always a pleasure.
Bingo.
Always a pleasure.
Absolutely. Yeah. Absolutely.
So maybe they're people playing bingo right now, having a barbecue.
Yeah.
Why
right?
do you think I said nothing?
Tweet their bingos into us.
You know, I put up that thing last week. Everybody can have it now.
That's right.
You got to stay near the mic.
I am near the mic.
Michael.
Thank you.
I'm Vincent Racaniello, and you can find me at virology.ws, which is 10 years old this month. I've been blogging for 10 bloody years. Not bloody. I enjoyed them. They're a lot of fun. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.
Was that a latent viral?
I was waiting for you to say bingo.